Y'all ever wonder how you're so different from your sibling, even though you both came from the same parents? Your sibling could be the pinnacle of humanity and the pride and joy of your family, while you crucified Jesus and sparked the Cold War. That's because of meiosis. Meiosis turns starter cells into sperm and egg cells, or gametes if you're a nerd, and makes each child that enters this wretched planet slightly different. Before meiosis, the cell starts out with 46 chromosomes, and after they go through meiosis and become sperm or egg cells, they become a haploid cell and end up with 23. Wait a second, why do sex cells only end up with 23 chromosomes instead of 48? First of all, Jason, shut your mouth and let me teach. Second, imagine if the gametes had 46 chromosomes each. If a sperm and an egg formed a zygote, the poor thing would end up with 92 chromosomes, which is literally impossible to survive with that many genes if you are a human. If each cell has 23 chromosomes on the other hand, the fertilized egg will have 46 chromosomes. Alright, back to meiosis. Before meiosis even starts, the starter cell must replicate its DNA. The cell starts out with 46 chromosomes and ends with 92 chromatids that make up 46 chromosomes. Teacher, you— Oh my god, Jason, let me explain, you hubris-filled sack of— See that thing in the middle of the chromosome? That's a centromere. One centromere counts as one chromosome in technical terms. So this and this are both one chromosome. Alright, now the technical stuff. Like mitosis, meiosis uses prophase, metaphase, anaphase, and telophase, or PMAT for short, to divide. Unlike mitosis, however, meiosis does this twice, so people label each phase with numbers. On the first phase, prophase 1, the chromosomes are getting, and they start lining up with their homologous pairs. A homologous pair of chromosomes are about the same size, and they contain the same types of genes in the same place. Now, when two homologous chromosomes get together, they do what is called crossing over. This is when they start trading genetic information with each other, like Pokemon cards or vital organs. This information exchange is a reason siblings can have very different traits from each other. During metaphase 1, homologous pairs of chromosomes line up in the middle of the cell. That's literally it. In anaphase 1, the centriole's spindle fibers grab onto the chromosomes and pull them away from each pair. Up next is telophase 1. This is when a nucleus forms around the separated chromosome clusters, and the cell is split into two slightly different daughter cells. Time for the next phases. Next is prophase 2, which is really boring compared to prophase 1. Notice in metaphase 2, the chromosomes are in a single file line instead of being paired up with another chromosome. Anaphase 2 is a little different from its first counterpart and pulls away chromatids instead of chromosomes. After that, the cell forms the nucleuses and splits for the final time, which creates either sperm or egg cells, depending on your sex. Because of crossing over and the fact gametes have less chromosomes, every cell is different from each other, and different gametes can result in a different child.
Oh, just the taste is interesting, but it's too spicy.
You're eating mayonnaise.